Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny. In a previous life, I dropped out for my PhD in microbiology. Then I was a fact checker and editor for pharmaceutical advertisers, and nowadays I'm a member and the president of Biotech Without Borders, a nonprofit based in NYC, dedicated in the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology. I've mostly worked in bacteria, but I've also worked as a research integrity specialist. I'm currently working for a scientific journal.
Every other week, we meet to talk about microbiology, and on a typical day like today, we do an overview of some of the coolest microbiology papers that we've seen in the last 2 weeks.
And every now and again, we try to do a deep dive into a paper to really drive into the core of the subject and understand exactly how the science was done. So if you see any paper that has caught your fancy that you really want us to investigate, message us at microtwjc and let us know.
Yep, you can follow along with the papers that we discuss on either week in our shared Zotero library. Our social media handles and the link to the library are in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us at microtwjc. And boy, do we have a show for you today. Firstly, we're going to be looking at how are scientists looking for where the next pandemic will come from. Will it be a spillover from a new virus from bats, or maybe another one from primates? Or what about diseases that we already know about that currently confined to one area but could spread to others due to things like climate change?
Mm-hmm.
And then we also look at the science of germinal centers and how if we vaccinate slower, that might create a new type of immune response. And also, what happens when bacteria mutate in space? We've got the hidden transcriptome of tuberculosis. Plus, we've got some mysterious things happening on that linking the cytoplasm to the outer membrane of bacteria. Lots of cool stuff happening with cryo-electron tomography. But first, we'll be looking at SARS-CoV-2 and how it evolves in response to antibodies. I'm just going to pull up the slides.
Yep. Yeah, the title of this paper, the first one that we're covering, is Mutational Induction in SARS-CoV-2 Major Lineages by Experimental Exposure to Neutralising Sera. So the authors are interested to know, if SARS-CoV-2 gets exposed to immunized serum, this happens in infection, during the infection process when people are infected. It could happen in the hypothetical, immunocompromised individual where tons of virus is able to grow up and sort of explore different variations. It could happen if you're given monoclonals or polyclonal serum as a therapeutic in the hospital. So there's many different ways a virus could find itself exposed to various neutralizing antibodies, and then they sort of track what mutations appear after those passages. But they're using cell culture in this particular paper. So they have viro cells that they passage over and over again. And they have a control where they don't add any serum, and they have a control because there's also mutations that happen just in adaptation to viro cells themselves. So there's also a control to see that sort of stuff. In this first figure that you pulled up, this is like the results of one of their experiments looking at the B1 lineage and culturing it with various neutralizing serum pools and seeing what mutations pop out.
Yeah, this is interesting because I do recall this because there are lots of kind of research going out there that are doing deep mutational scanning, trying to look at all the various kind of permutations of the spike protein.
Mm-hmm.
And this is an interesting approach to get insight on the kind of evolutionary arms race that's happening between SARS-CoV-2 and us at the moment.
We know that the mutations will definitely, mutation is always happening. And so having, as we become immune through vaccine or through infection, and as long as there's continued spread of the virus, there's going to be variation. And picking out the patterns, the authors are interested in picking out the patterns that they find. And Faz, you mentioned we had been covered these deep mutational papers in the past where they made every mutation inside of the spike protein to see what would happen. But one of the other things in this paper, which is nice, is that they're not just looking at spike, they're looking at the whole genome. So we've seen a deep mutational scan of just the spike protein, but here they can look across all the different proteins to see where mutations occur. I think that they don't see, they see a few mutations that occur in other spots, but that aren't the spike. So like that's kind of a curious observation to say that there are some mutations that are happening outside of the spike region, but they don't really point to. I didn't think they did a good job of pointing out to, or at least maybe I didn't pick it up from reading the paper, pointing out like what the significance of those things outside of the space were. They do say that several of them are not significant because they're just like the background mutations that happen right as they're being passaged. Yeah, they spend a bunch of time talking about ORF7 being deleted away, or like the deletion of ORF7. But the issue is that they're doing this in a cell culture, so like that cell culture system doesn't have the same immune response that like a full organism would have. Like it doesn't even have. I think it doesn't even have like the interferon, like a strong interferon response, which is just like the intracellular immunity, if you will, the innate intracellular immunity. So like seeing a mutation in an immunomodulatory like object there, it could just be like it's not useful to make in this particular model system, and so that's not particularly insightful about what mutation would be like in our context, in the whole organism evolution context.
Yeah, and I think as we mentioned before, viral cells are known to like cause SARS-CoV-2 to like lose genes or lose kind of things that would be very important for infection in the normal host. So one of the classic things is a furin cleavage site is the first to go in viral cells. But I think that there is an interesting opportunity with this technique in terms compared to deep mutational scanning. They just make a mutation of all of the kind of the SARS-CoV-2 kind of areas, and then they throw out an antibody to see whether it works or not. But that's not exactly how evolution works, right? Because evolution is iterative. So each mutation can create opportunities for other mutations to be viable with the original version.
Yeah, absolutely. Yeah, and so here, like this gives you like maybe closer to the what could happen, and then of course like you can grab these observations and try to compare them against like what mutations we have seen emerged, right, in the lineages that do catch on. Yeah, and I think that's all I really want to say about this one. I don't think in reading it, I didn't really take away too much. Like I don't have more, at least like, yeah, for me, like reading into it, like it seems like this paper could be helpful in like a series of papers that like sort of talk about immunity, maybe blending in some of the observations that we've seen from deep mutational scanning, or like more about like, I don't know enough about the specific mutations. Like, I feel like a lot of this mutation world, like learning about which mutations are interesting, you kind of want to talk about specific mutations because those are the ones that's essentially what you're, that's what you're studying, right? Like you're trying to see like which ones are the important ones and when do they occur inside the lab versus when do they occur inside of the general population, and like using that to give you a sense of what could happen in the future. Yeah, I was like, I thought it was kind of interesting from this perspective.
Yeah, I kind of agree. It is like a good first chapter, but there is more that could be done, and yeah.
And there's severe limitations to like sticking inside of cell culture to do this evolution experiment, right? Or like doing any sort of in vitro evolution and trying to say like, how does that relate to the greater population? Just because of the complexity of the environments that the greater population sort of represents, rather than the controlled like experiments in the lab.
Yeah, I mean, it's literally just one sample of neutralising sera, so that's one aspect of the immune response they're responding to as well. But there's a lot more complexity there that could be built upon. So I think that this could like provide some insights, but I think more it's kind of a good start, perhaps.
Yeah.
But yeah.
Yeah, and I think again, like where I'm taking the value from this is that, like, potentially we could bring together, like we're not doing it right now, but we could bring together like this paper along with some of the other stuff about mutations and come to like a better understanding of the whole list of like mutations that occur, like which ones are useful and why we think they're useful in different contexts. All right. Well, let's move to the next one.
Next one, we're moving away from SARS-CoV-2 quite early, but we're looking at primate hemorrhagic fever virus causing arteriviruses poised for spillover to humans. So primate hemorrhagic fever is a very terrifying thought because Hemorrhagic fevers that make you bleed a lot. Not great. But this one affects primates mostly. So what they looking at specifically in this paper is Simian hemorrhagic fever, which is a lethal disease that has been spotted in captive Asian macaques. And outbreaks have been occurring in macaque colonies worldwide for a while.
Yeah. I think that was kind of
yeah.
spooky to read in the introduction that this is like a well-known thing that causes outbreaks within within Asian macaque colonies. But I guess there's enough. It's well known enough that there's a lot of controls. It sounds like to make sure that these outbreaks are controlled, are contained properly.
Yeah. So the thing is, they don't quite know whether these can spread to humans because humans are primates, so there is always going to be the risk there. But so far, it's affected macaques, which are monkeys. I think there is a distinct difference between monkeys and apes that I'm not qualified to say.
Yeah.
But also the tricky thing here is that this is mostly a very difficult to culture virus as well. So in order to culture it, you need to find the host. But of course, this virus is lethal, and a lethal virus tend to find it in a dead end host. So a host that dies immediately, it probably isn't the original host. So you'd want. So I think that they tried out a number of different like cell lines. I think they got to grow in Grivet cell lines. So Grivet is an old world monkey that looks like your standard monkey, and they. Yeah, I looked up on Google Images, like, yep, that's a monkey. Yeah, David Attenborough weeps. But yeah, so what they did was they hypothesized that CD163 might be the cellular receptor, and they knocked it out in single cell clones of like this Grivet cell line to see whether it stopped the virus from causing infection, and lo and behold, it did. So that kind of indicates that CD163 is an important receptor. One thing to note is it's an intracellular receptor. So that means it's inside the cell. So the actual virus must be finding some other means to get inside. But in order to get its lifestyle like kicked off, it needs to have that intracellular receptor.
Yeah, I mean, I think that's like it's pretty good sleuthing to already nail down at least one molecular determinant, right, and then be able to screen through like. It's like the beginning of getting like a really good like a molecular surveillance thing, even though it's only one step on that chain. Like that's enough to at least start to say like, is it possible for this thing to get into humans? Yeah, like they know one step of the chain. They can do it. I mean, they do it too, right? They do a deletion of delta or of CD163, and then you can complement it back in. You can put in any copy of CD163 in from a variety of different organisms and try to get a sense of like what is that, what is the right molecule for the job there, or like that makes the cells permissive.
Yeah, and that's what they did. They tested out a number of different monkey species at first to see which ones are permissive for the infection. And what they found was that yeah, rhesus macaques were permissive, and maybe not all the monkeys. So then they turned to apes, so they found that like SHFE can replicate in the primary cells of a chimpanzee, a Western gorilla, and of course the most important primate to me at least, humans as well. So they found that the human CD163 ortholog could also support like Simian hemorrhagic virus replication, SHFV, I'll call it, in these cells. So they're not testing cells; they're testing the actual, just the version of it, CD163, and that's within the Simian cell. So the idea is that if the virus could potentially get past all the other things, it could hit humans.
Yeah, they do some human cells as well.
Yeah, they do. Macrophages. So one of the things that they notice is that there has been previous studies on this where they test out lots of human cell lines to see whether SHSV can infect them, but they found that it couldn't. But one notable thing about those cell lines that were chosen was that they didn't have much CD163, which is that tends to be found on kind of monocytes. I think monocyte precursors. So they looked at kind of the precursor cells and found that it could infect those. So it could replicate in those specific human cells, which means it could potentially spill over into humans at some point. So,
it's like This paper is like a warning. It's a warning because, well, it's a warning, but it also is, it is very careful in picking out specific scenarios in which they see a permissive environment for the replication of this virus, and then the warning to us, I suppose, as readers of the paper is, like, because this exists, because they can pick out these scenarios where the virus can replicate in places, in ways that seem close to our organism, we have to watch out. We have to make sure that we're watching for outbreaks of viruses like this, because, yeah, I mean, this is the idea of spillover from a zoonotic source and hemorrhagic disease. That's Ebola as well, right? That's the same sort of life history that we associate with Ebola. So yeah, I guess there's another family of viruses that fit in that category, and they deserve to be watched as well, because yeah, in an increasingly globalized world, like these are the potential threats that exist for us.
Yeah. I mean, I'm going to say that there does need to be a lot more work to establish the rest of this virus's life cycle. But I think what this shows is how important it is to keep an eye on viruses like these, which are affecting our primate cousins, or have that potential capacity to spread over to humans. Because I think humans are contacting the natural world more and more frequently, and the amount of time it would take me to get to the Amazon or like these other areas that might have really scary diseases, is becoming a lot shorter as humans increase their contact with those kind of environments.
Yeah, yeah, or just like increasing contact in general with people like all across the world, right? It's like the degrees of separation that you have between you and the jungle is getting smaller, and the jungle is full of life.
Yeah, and yeah, so that's one kind of potential spillover virus that we maybe should be worried about. I don't think we should be worried about it too much, but that's because I'm like the mayor from Jaws. Here's my spirit animal. Why not just enjoy the beach? So what if you get eaten by a shark?
But I think maybe it's not about like being afraid personally, but maybe just understanding that viral surveillance is like an important part of like ongoing life on the planet. Yeah, and like I thought it was a really interesting paper from the standpoint of like how they came from like a observation of disease in monkeys, and then they start saying like, well, shouldn't we also check for this in humans, and how would we begin to do those checks, right? To like start to build the case that this could be something dangerous. And yeah, like I just like that was very interesting to me to read that logic, because I mean, I don't get exposed, I think, to that thought process very often.
Yeah, I think there is an aspect of trying to figure out what is going to be an important virus or not, and trying to justify that and show people that maybe we do need some more funding in this area to make sure that to check whether these are threats or not.
Yeah, yeah. And then what should we check it, right, to be threats? And like that's sort of to your point, like they were saying, like they have this one particular thing, CD 163, but like could there be other things that we should be looking for? How do we even find those things, right? Like they sort of start with the knowledge of like a factor that makes these viruses permissive. Like think about scenarios where you don't have that starting point. It would be a lot more difficult. Yeah, anyway, that's not in the scope of this paper, but like when thinking about viral surveillance, I think that that's another issue that sort of emerges.
Yeah. Speaking of other viruses, an ACE2-dependent Sarbecovirus in Russian bats is resistant to SARS-CoV-2 vaccines. So this is another potential kind of keeping on the theme of potential spillovers. This obviously might seem a very familiar idea because SARS was also a spillover. We don't know where it came from, I don't think specifically, but there's plenty of arguments on Twitter if you want to.
Yeah, I think people agree that the one before the spillover one was a pangolin, but the one in between, yeah, people aren't sure.
Yeah,
I think bats though. Bats is one of the forefront hypotheses.
Yeah. And so this paper focuses on a virus was discovered in late 2020. So in late 2020, there are a couple of viruses which were discovered in rhinolophus bats in Russia, and they're called Costa 1 and Costa 2, and they kind of looked into the So when you have a new virus, you have to figure out what's related to, where does it sit on the family tree, and figure out. And in this case, they realized it was a SARS Sarbecovirus, so that's a SARS coronavirus kind of family. And they looked into like kind of to its relatedness. It's actually like fairly unrelated to SARS-CoV-2 compared to say MERS or other viruses in the family. And they're obviously very interested in the spike, which is the main way that these viruses are used to enter cells, and what kind of is a surface receptor uses. Because I know that with MERS it's DPP4, with I think, but with they found this was ACE2 dependent. So again, it's also got that. There's a lot of similarities to SARS-CoV-2. So they wanted to check whether it could enter into human cells. So that's kind of what this paper is looking into. Again, in the same way to the last paper, trying to figure out what the threat of this virus is.
Yeah, and I mean this one I feel like they don't go deep into like whether or not it can replicate inside of cells, because they're using like I think they're using
VSV, vesicular stomatitis.
Yeah, VSV.
Virus.
Yeah. So they're really just interrogating the receptor and the receptor binding sort of pair, rather than the whole virus itself.
Yeah. And I think that's kind of an important thing to have in mind with these studies, because for a lot of these studies, they're not looking at the kind of, for the last study they weren't looking so much at human cells, they're looking at the human cell receptors. And in this one, they're looking at the spike protein specifically, and so there is a number of reasons for that. I feel partly it's because
you got to isolate things too, right? Like it's easier to work with just the spike, because then you don't have to worry about the biosafety concern, right? Because you're just looking at the proteins on like a membrane surface, pseudotype particles.
So it allows them to work in a class 2 lab, because class 2 labs you have gloves and like kind of lab gear. Class 3 labs are a real pain to work with, because you have like kind of the full like outbreak sort of kind of deal, and it's really annoying to work with.
Yeah, like if these pseudotype viruses got out, I mean, it wouldn't be a good thing, but like if the pseudotype viruses got out, like the expected danger of them is really just, you might maybe some genotoxicity, right? It might like make someone express some weird protein or something like that, but like maybe not even that, right? Like the payload inside is not like considered like a dangerous infectious payload. But of course, like the proteins on the outside, they may facilitate fusion of some sort to the membrane.
So he looked at the spike and found that it can, and so I think it can enter into human liver cells, but as long as there is like a trypsin or some kind of protease that can allow it to enter. And
then I guess the danger that they're trying to describe, I don't really understand where that danger is necessarily, but they're sort of worried maybe there could be recombination between this and SARS-CoV-2, just because they have that similarity. Like maybe it could get picked up, or maybe they're also worried that this will become a virus that will infect humans. I'm not really sure. I couldn't pick it up there.
So one of the main things they point out in this paper is that it's very resistant to serum from SARS-CoV-2 vaccinated individuals, and it can still bind to ACE2. So it means it's binding in the two. And I think they are concerned over potentially like that receptor binding domain being like crossing over with the SARS-CoV-2 version, and then creating an entirely new receptor that is resistant to all vaccines. I think that is part of it, because I think that they can't really say that this virus will spill over, because they're only looking at this very narrow aspect of the virus. But
they don't know if what's inside of it is enough for it to replicate inside of human cells, right? They haven't done like what we saw in the previous paper, where they actually tried to use cells from the potential host.
They have only like one step of the process, and there's a lot of other complicated steps of the infection process that we don't know whether this virus could work in humans or not. But they're focusing on the main first part, and the argument that I think that it is a possibility, but
but it seems rare to me. But again, I'm not really sure like what that where
it
that
really
lies.
Depends on because I feel like if we infect the species of bats with SARS-CoV-2 virus, then it becomes a much bigger possibility.
Yes, yes,
so
yes.
And we've seen that with a lot of animals where like humans are spilling over SARS-CoV-2 into like deer populations and ferret populations and other places. So if we manage to give SARS-CoV-2 to these bats, then that could be a bit dangerous.
Yeah. Okay. No, I think I really like that. That's the right way to think about this. I think, right?
You
It's
like that? I hate that.
Oh, I mean, no, I mean, it sounds horrible, but that when you said that, I'm like, oh, that clicks better in my head. Being like, I'm like, where's the risk here? Like, I was trying to understand, but it's very much like, yeah, the spilling back into the animal population, right?
Yeah.
Yeah.
Because like as it stands, we don't know whether the COS2 will ever spread out into humans, but we know that SARS is being carried by humans to all areas of the globe.
Right.
So
yeah,
we don't know what that will do to other native coronaviruses that are in different species.
Yeah, and where those viruses, if they do spill into animal populations, where they will find sort of inspiration, right? Or like genetic genetic variation that would make them more adaptable in the context that they came from, which was us. Yeah.
Yeah. So
yeah.
Let's move on to another paper that is also doomsday-like, but less doomsday-like than this, because I feel like I've brought to being just by mentioning that. But just, it's like if you mention a terrible scenario, it's more likely to happen. It's like, oh, great. Why did I say that?
Voldemort style. Yeah, the title of this one is Predicting the Evolution of the Lassa virus Endemic Area and Population at Risk Over the Next Decades. So this one's doing more of, I would say, well, it's definitely, they're building a computer model based on data. They're taking a more zoomed out view. They already know that Lassa virus is sort of a potential emerging threat, biosafety threat. It's another hemorrhagic fever, so one of those frightening Ebola-style viruses. Yeah, and they take data from the history of different outbreaks and I think especially like geographic location of where these things have happened. And they build a model that captures also the environmental factors. What did it say? Ecological niche models considering temperature, precipitation, 7 types of land cover, human population. So they have all of these factors that go into explaining the occurrence of Lassa, and they want to make predictions using the model.
Yeah, Lassa virus is interesting because it's carried by a rodent that has a certain lives, and even though Lassa is often found in these rodents, it's not found in all the rodent populations. So part of the thing that this they have to model is where these rodents are likely to appear in, say, over the next 100 years. Like how is climate change going to affect the where these rodents are going to be? How is it also going to affect the Lassa virus within the rodents? Because I think one of the big unknowns at the moment is like, why do some areas have lots of Lassa virus with these rodents, and why do some areas of these rodents, which I think the name is Mastomys natalensis. I'm not going to say that all the time, so I'm just going to call them the rodents. Where they in some areas they don't get it, and because I think the rodents are found anywhere where there's like high precipitation, but Lassa virus itself it needs to have like certain like areas, like it needs to have some like specific land cover preferences and and temperature kind of ideals. So the things they need to take into account is like what makes Lassa virus more likely to spread, what makes the rodent more likely to be susceptible to the Lassa virus, and also the human populations within the areas affected by the Lassa virus. So because because humans aren't standing on their own, our population is going up. So within the Lassa virus's current range, you've got lots of places that have increasing population or like increasing economic wealth. So there's been a recent increase in Lassa virus outbreaks in Nigeria because that's undergoing quite a lot of development at the moment.
Yeah, and I feel like so their model or whatever, they're able to take it and see that it replicates, I guess, these very logical understandings of like the linkage between these environmental factors and Lassa outbreaks. So yeah, so I guess like to me that's that's the strength, I guess, of the paper, is that it gets to reveal to us in sort of more quantifiable terms, like what of these factors would influence outbreaks, and part of that is dependent on growing population centers being like those places. The virus is endemic in places where there's growing population. So that means the risk will increase over time because we expect more people to be in those places that are risky.
Yeah, and those people are more likely to travel out, and luckily, since people don't spread the disease, like we're not worried too much about a pandemic. The thing that we're worried about is.
But it's concerning for the places that are that this disease is endemic, right? It's like it's saying that in these places, like there needs to be some attention paid.
Yeah, it affects those places, affects like the development of those places, and I think, but what they found was quite interesting because, again, they don't know the specifics of what causes the host of Lassa virus to be infected, but I think they have a few things to point towards it, and that's kind of interesting. And they also like find that once like Lassa virus gets into a new area, it takes a lot longer to take over the host population there than you'd expect. I think they said like it travels between 0.8 and 1 kilometer a year. So actually like not too slow. That's still pretty it's not
for a tiny virus, but like a tiny virus that is in the body of mice.
Yeah. So that kind of like that's their almost estimate for how much it'll spread through the rodent population and how long it'll take for how the climate will affect that. So once like say you get more areas with like higher rainfall, you see the little mouse thing going out into all these new areas, then the virus will follow it at this kind of 1 kilometer per year pace. And that will be that kind of having that idea in the model might be useful for people who are planning for this in the future, and then it might help feed into control strategies.
Yeah, and I think this paper is one of those is like kind of important on the side of just arguing for surveillance for this virus in these areas, and also that these areas need like healthcare support, right? Like how do we like I think they pointed out like there are no like vaccines or there are no vaccines for Lassa right now. So like it's also like adding importance to this because like they're candidates, but like they need to be focused on so that by the time that we see a more widespread range of this virus, the places that are going to be affected have the tools to deal with it.
Yeah, I mean the only vaccine candidate that is INO 4500, which is an Inovio vaccine, where
DNA-based
DNA-based electrocution. This is my least favorite vaccine. I don't. So yeah, hopefully we can find a better vaccine for it. But in the meantime, we need to have better infection control.
That's all I want to say about this one, I think.
Vaccinations and immunity. Long prime germinal centers with enduring affinity maturation and clonal migration. So,
really, this is a deep immunology paper. This is a deep immunology paper, but I feel like there's relevance because we've on this show we've talked about germinal center B cell responses in the context of SARS-CoV-2 infection and how SARS-CoV-2 can mess it up a little bit, so you don't always get the best virus, the best B cell maturation. So here they're just studying this B cell germinal center itself and really trying to find a better vaccine strategy. And so they have a specific cell type, like. When we talk about immunology papers, we're often fixated on like a specific cell type that's useful in a specific immune process. So here they're focusing on germinal center B cells, which are B cells that have been selected, that they have the antibody on their surface, and they're being selected to expand, but also to have slight variations so that they might get better at recognizing that antigen. So like from their original recombination, they've been picked out of that crew, and now they're going through iterative phases of expansion and selection to find even better versions of themselves.
Yeah, and I think from previous work, we know that like that evolution continues to go on, even quite a lot longer after, like, say, vaccination and the infection's resolved. Because I remember there's like a pivotal paper we looked at where the antibodies were still like evolving to become slightly different even after the infection was happening. I think this paper is following on from that, although it focuses in on HIV rather than SARS-CoV-2. So, and they're kind of testing a new vaccination strategy. So the idea of slow vaccines, which is instead of say giving one shot dose at one time, having kind of a longer slow release of this longer time period, almost mimicking a long-term
Wait, you say
infection.
slow release, but I think what's implied is that you get multiple vaccinations over the course of a long period of time. It's not like a single dose that like.
Yeah, I think
okay.
I think in this study they work on rhesus macaques and they multiple vaccinations over a series of time points, and they have like so they have like one control of so they're using HIV as the test for this.
HIV like a specific antigen from HIV.
The envelope trimer. Which is an important because HIV evolves very fast. The envelope trimer is changes quite quickly, and I think one of the things that is quite pernicious about it is the envelope trimer. So I think this happens with a lot of antigens, is that the antigen that is most likely to create an antibody response is also the one that varies a lot. And I think we see
it's a good evolutionary strategy on the virus side, exposing that as the immunodominant, because then it means that it can have a second chance to get in after some time has passed.
Exactly. So it's in that virus's interest to make sure the immune response to this certain variable aspect of it. I mean, we get the same thing with the SARS-CoV-2 S2 portion, where again that's very, very conserved, but also very hard to create an immune response against. And so what the idea with this long vaccination is to test whether you can, having a longer time would give these antibodies a lot more time to evolve and develop a response to the antigen.
They have the observation that B cells mature over a long period of time. So then the investigators go into that thought process saying, how do we take that principle and design a vaccine strategy around that so that we're like working with the body's natural way of like reinforcing the B cell population and having it like, just providing that stimulus so that it's continuing down that path to make better antibodies.
Yeah, so you get one group that is just given one big like bolus injection, another group that's like injected like regularly over a period of time, and they measure the antibody responses, and I think the germinal center specifically as well, to see to check what kind of antigens what kind of antibody response you get out of it, and also like things like how quickly that response declines. And what they find is that the ones with most priming tend to have a much higher antibody, like levels of antibody just throughout the whole. So the decline factors tends to be a lot less. And the thing that I'm quite interested in is that the scope of the antibodies, the antibodies cover a lot more of the antigen. So I think it's most visible in G. Where they kind of show that for group 1 is the original one, where you see like only maybe 1 or 2 epitopes on that protein tend to get a lot of antibodies attacking them. But with group 2, group 3, you get more coverage over different parts of it. And that might include the areas which are less likely to evolve changes. And
this figure G is fascinating to me because they're using electron microscopy as like a screening technique. Like these are electron micrographs they have where the gray is that enveloped tri piece or whatever, and then these like the colored blobs they have hanging off of it, those are fabs. So they take the polyclonal serum and just take the one little portion that binds to things, and then they wash them over a whole bunch of these different envelope proteins to see what binds. They take that and they put that under the microscope to look at it. I mean, not a regular microscope, an electron microscope, because they have to look really small. But yeah, it's a funny method to go and look at the diversity. But it also generates these wonderful images where it's like, it's so clear. Like every image, you're like, yeah, that's one protein, and there's a bunch of fabs attached to it.
I mean, I love this because like usually you'd pull out a sequence, maybe like the protein sequence that the antibody's protecting or specifically binding to, chop it up, and then you have to like go back and do lots of molecular modeling to figure out where it's attached to.
Right,
Here, they just
right.
look at it immediately, and you can see immediately, oh, the antibody's attached to that area or that area, and so it's very direct and very
Yeah.
visual.
I think it speaks to how far, right? Like we always talk about cryo-EM and how it's like changed all this stuff, but like I guess the same, it's the same visual techniques that are being used there are inside of regular EM as well. Like I don't, maybe this is probably a precursor, I bet, to that complex thing, but like it's nice to see those technologies are going hand in hand and how scientists are getting used to using them inside of their workflows to show interesting phenomena.
Yeah, so I think this is really interesting in terms of getting that broad antibody response, because I think we've mentioned before that the more area on the kind of antigen, the protein of the virus that you can hit, the more you constrain its evolution. So in order to escape, it doesn't have to just make one mutation, it has to make lots of other mutations. And that makes it much more harder for it to avoid the antibody response. And that's very important for fast evolving viruses like HIV, where you really do want to constrain that evolution as much as possible. But yeah, it can also be useful for things like SARS and other things. But yeah,
I was just gonna say, but certainly like this immunizing strategy seems very burdensome to like a person to have to undergo, to be like poked repeatedly over the course of a month or something like that.
That is the one thing. So this is not practical in any way yet. This is still very much in a lab. I mean, but there are various things that people can think about it, because like just having a vaccine implant instead of a vaccine. Again, that's a bit further.
Yeah, you're imagining the future tech, right? Like maybe in the future it will be something that's like slow release of these antigens.
Or even like thinking about like a liposome with slow release or something like that, which.
I actually, the first thing that comes to mind, and this is just like some random connection that I just thought of, but like there are some like hormone therapies that you get, right, that are intradermally, like they're subcutaneously injected, and then they just sit there and then they slowly dissolve into you, and so like they give you like release over like a month. Anyways, it's possible in chemistry. They do a lot of stuff like this.
Yeah, it's definitely possible, but I do think there'd be a lot of pushback if you, because
yeah, for sure.
Because like
it's a hard technology. It's a technology that has to be deployed in this specific arena as well.
Yeah, I think it'll only really work for like things like HIV or things that are very like terrifying diseases that don't have that kind of vaccine yet. But I think in terms of understanding how the immune system works, I think this is a very important and interesting thing that could like lead to other strategies.
Yeah. Okay. Well, that's all I want to say about that. So we can move on if you're good with that.
Yeah. So discovery of a novel inner membrane associated bacterial structure related to the Flagellar type 3 secretion system.
Yeah. So I thought this is a funny paper because they just talk about looking at a lot of these cryo-EM images and noticing that there's a structure that exists in a bunch of them, and it looks kind of like a hat is how they describe it. Yeah, that's a good picture of them. It looks like a hat, and they end up finding it in a whole, they originally find it in some subsection of images. I think they're looking at Helicobacter, maybe pylori is the first one. But then they expand that, and they're like, well, does it exist in all these other types of bacteria? And they're like, oh, it's all over the place. And they do find that it's dependent, at least in the Helicobacter pylori sense, on a specific gene, FLIP. It's like they find a natural point mutation that disrupts it. And that becomes their clue to sort of go in and ask, like, what is this for? Because that gene is like pillin related, and so they think that it's part of a secretion system for. They think that it might be part of some sort of secretion system related to that. Those pillin, or I guess flagella, or similar, could be derived from similar structures. Yeah, and I mean, actually, that's all. I feel like that is the paper, is that they saw a picture And they notice this structure in some things. They have natural variant mutants that don't have that structure, and they can infer some. They can infer and they can also replicate, right? Make those same point mutations in different backgrounds, see if the structure changes under EM. So it's very much just gene to electron micrograph sort of paper, but it's a discovery because like this particular complex is not sort of a well-described, like it is described in some contexts, but not described in many others. So it really is giving, I guess it's like it's like another secretion. It's another secretion system to consider, and one that has sort of a specific role because I think they look at they look at whether or not if you need this structure to make the pilus or the flagella or secretion of proteins. And they find that you don't need it to secrete things, I think, is what they look at. But you do need it to assemble like the beginning of the flagella.
Yeah, so I think it's interesting. But yeah, this is like we still don't quite understand what the structure is or what it does. But I think the idea that it helps, it's kind of a scaffold to help build things. It's really an interesting idea.
Yeah, because they have this image that obviously has density, and they know that some point mutations break it, but like there's so many proteins that go into that little image, right? Probably there's many proteins that go in that image, but we don't know what they are. But we know that they come together, and it looks kind of the same between bacteria. Yeah, I find this has like a weird analogy to like when people study like the beaks of, right, the beaks of finches or something like that, right? Like you see all the finches have beaks, and maybe right? Like there's some weird ones that like they have a certain like lifestyle or mutation that like makes them look a bit different. But this is on like the really tiny scale. Like we're not looking at beaks, we're looking at like the cell membranes and specifically structures in those cell membranes. And they sort of, we can see that they're in a lot of things and we know like some of the dependencies, but there's so much we don't know already. It's so much a raw discovery, this paper, I think.
Yeah, so what does the bacteria hat do? We don't know. We'll find out.
Yeah, what makes up the thing, we don't really know. We'll also find out. But again, like the small clues that they do have is that it's not for regular secretion. They can still see these cells like leaking out proteins into other cells. When they make mutations in bacteria that could inject proteins, they still can inject. So this is not like this structure isn't for injecting, and but it does seem to be for advancing like some of the first structural components to build up the early flagella. Is the one effect that they do see, right, in some of the mutants that they have.
Right. I think that brings us on to the next paper, which is also related to the membrane. A multi-domain connector links the outer membrane and cell wall in a phylogenetically deep branching bacteria.
Yeah, so yeah, another cell wall thing, another thing that they use electron microscopy to look at. This time they have a bit more of a specified question. They're looking at Deinococcus radiodurans and saying that it has like a kind of a strange cell wall situation going on where it stains gram positive, meaning that the idea is that, it stains as if it had this huge peptidoglycan layer, but actually when you look at the cell wall, it has a little bit more of a gram-negative style cell wall, where it's like a thin peptidoglycan, and then there's like a outer membrane, and then there's like some stuff on the outside. So it already has like a kind of strange surface. The reason for that is it's a very old bacteria in terms of like when it last diverged from the last common ancestor. So they call it deep branching, is like the term that they use to describe that. So it's kind of had a lot of divergent time to do its own thing, right? to create its own systems of stuff. And one of the things that it has is like proteins on the outside of the outer membrane. And I actually first heard about this layer. They call it the S layer. I've heard about this layer in the context of bacillus. So a lot of gram-positives, or some gram-positives rather, a large group of gram-positives have these outer protein layers. And yeah, and so there's a lot of structural similarities to this one. The SLH domain, that's a structural similarity that also exists inside of some of the bacillus versions of the S layer proteins, or like the proteins that form that layer. So I guess that's a good analogy for them as well in this paper to say like, oh, this is kind of like those proteins, and they use cryo-EM to dive into that structure. I think Faz, you were on an image of that structure a bit earlier.
In fact, they've got a nice little video that we can look at.
Ooh, nice.
But yeah, it's interesting because I think this paper is mostly diving into that structure and looking at its functions, right? And here we go.
Yeah, and seeing how it. Yeah, how part of it is like smaller. Has this like a smaller binding domain, right? That binds it to the surface of the outer membrane, I suppose, in this circumstance. And then on the top of it, there's just like a huge chunk of beta barrel, a huge chunk of protein, but that helps form the surface. And I think they do show, though, that it's not a contiguous surface on the surface of the on the outer part of the bacterium. Like, even though that's a really large chunk, there's lots of porous ways of things getting through. And if they make mutations in it, like, you get weird membrane vesicles. So like this protein is also important in helping form the membrane properly of radiodurans, the outer membrane, not the inner membrane, I don't think.
Yeah, I mean, I think that's basically all I have to say on that paper. I don't know about.
Yeah, just another. So this one, like, in some ways, this is like a more zoomed in discovery than the last one, right? Like the last one was like, it's like the pictures they have are of a complex that sits on the membrane. Here they like zoomed in. It's actually one protein, a really big protein, but yeah, just like this one big thing that sits on the surface, and it's in a lot of other bacteria, meaning that if we learn about this, we may also learn about a whole bunch of other things. Yeah, and the surface of bacteria really is a lot about it's their body, right? Like that's all they are. They're just the surface, really, and the stuff that kind of falls off of it. So yeah, so it's important to be able to learn about this stuff, these structures, because it really tells us about how bacteria live and like what goes into that life. Okay.
Yeah, right. So the next paper is on the transcriptional regulator. It's called the transcriptional regulator CtrA controls gene expression in Alphaproteobacteria phages, evidence for lytic deferment pathway. So this is an interesting paper because it's not like fully all there, but well, how can I put so the paper starts off as a description of a new virus that was put. So let me like go through my notes, because I read this paper and I read the abstract, and that's kind of what interested me. Then I delved into it a bit more, and so the main focus of the study is this VB underscore B sub S underscore like delta, which is a phage of Brevidomonas. It was discovered in like a freshwater pond.
Yeah. If you scroll us to figure 1, we'll see like a EM image of it. So yeah, very much starts the story as just a new virus, a new virus they saw.
Yeah.
which I mean, there's tons, right? Like there's like so many new viruses. So that in of itself may not be intrinsically interesting or like super novel.
Yeah. So they looked into the genome of it, and they found that there was some like aspects of some aspect of the genome, it looked like it bound CtrA, which is a cell cycle regulator protein that's found in a lot of different bacteria. So the idea is that during growth, CtrA would regulate the growth of bacteria. But also the idea that a phage has this site might mean that the phage is also bound by that same cell cycle regulator.
Yeah, I think that's really cool, actually. And like when I read it, that was really exciting to me, because I think they talk about it in the introduction as well. But the classic phage stories we have, right, are phages making the decision to be lytic or temperate, whether they should integrate into the genome and sort of wait it out, let the bacteria do its own replication and therefore replicate more of the phage, or whether they should burst out and just like start lysing everything so that they can just release more of themselves into the environment.
Yeah, and that's an important. So I think the other thing they noticed in this paper is that they looked at the phylogeny, looked at like other phages that have this sequence, and they had this group of phages called the LPG group of phages, and that's where the paper shifts to be more about the whole group of phages rather than just this one virus. And they
Hmm.
looked at kind of the how often they have CtrA binding sites and what that could mean. So one of the things they noticed is this group has a lot of they tend to be flagella binding viruses. So bacteria have like these flagella that they use to like swim around, and these phages bind to them, and
Hmm.
that's very important for infection. So of course some bacteria don't are both like have flagella and also can sit down and lose their flagella. And the phages really want to make sure that when they spread, there's lots of other bacteria out there which have the flagella. So the authors speculate that the CtrA binding site will help the phage know when other bacteria have flagella in order to
Yeah.
enter that lytic phase. Um, they,
Yeah.
but
They don't know for sure, but like that's the hypothesis that came out
that's
from this.
yeah, that's the
Um,
hypothesis. But the only evidence they have at the moment is that there's a lot of CTRA binding regions, and
yeah.
yeah, I think that's the main thing. So
Oh,
again,
and
that is
I think the other evidence is that CTRA does bind these sites and like upregulates some gene expression in the
yeah.
phages
Oh
or
gosh,
downregulates gene expression. Yeah,
yeah. No,
they have a little bit of like, oh, there is interaction, so they know that it is like it's synthesizing this signal somehow. They don't know how that signal like ultimately gets processed, but like yeah, it is binding and yeah, it is changing some gene
yeah.
expression.
I don't want to shame their shift assays, so I'm gonna go. But yeah, that's the main sort of evidence. So I think it's still very early on. I
Yeah.
think that it's called like a letter rather than paper, where it really has like the initial bits of a hypothesis, but doesn't have all the evidence to back it up yet.
Mhm.
Um,
Mhm.
but yeah,
Yeah.
but I still think that the concept is very interesting. Um,
Absolutely. I think obviously, or like, I think for folks who study a lot of biology, like we always know, like you find one example of a story and that could be like an exception sometimes, but like really there's so much diversity in the world that like in ways of living for natural organisms or for biological organisms that you might find like similarities in another system, right? Use sort of some of the abstract concepts. So here I really like that it's a totally different system, right? Almost a wholly different like type of phage and lifestyle that you could imagine from Lambda. But here it has like a similar sort of feedback mechanism where it wants to watch a specific like transcriptional regulator inside of the bacteria inside of their host in order to find the moment where it could work. But again, like that story needs to be fleshed out. We need to like keep asking questions about that story, making more hypotheses, getting more data, and then we could finally, write that in a textbook and talk about that more widely.
yeah, and I think it's an interesting concept to think about for human viruses that are dormant for a long time and then have a trigger. So sometimes that
Mm,
trigger is that the immune system is slightly depressed because you're stressed out, but there could be other things.
yeah, all that Epstein-Barr stuff, right? Just hiding away, waiting for the moment.
Yeah. Uh,
All right, next one we got is
Non-random
non.
genetic alterations in the Cyanobacterium nostoc species exposed to space conditions.
Yeah, pick this one out because I don't know. It's like so clickbaity, but also like I want to know what people are doing in space when it comes
Yeah.
to biology. Um, in this case, it's like a 23-month stay on the International Space Station. They send up a specific species of cyanobacteria, nostoc. Um, Cyanobacteria, they grow in like these big chains where they actually have like differentiation inside of them. So like part of the chain does photosynthesis, but the other part is like impermeable to oxygen and does nitrogen fixation. And so they're really cool. Colonial, responsible for making tons of oxygen on our planet. Thank you. In this instance, I think they're really imagining these cyanobacteria as some sort of like mini terraforming agents. They want to push them to their limit and see like whether or not yeah, they could adapt to harsher environments because there is some evidence to suggest that they've adapted to like deep subsurface conditions or like some sort of like increased radiation damage. Like there's history on the planet Earth to know that these organisms have adaptable capabilities, but can they adapt to space? So or space-like conditions. So they have this strain of nostoc, and they send them up into space in a bunch of different conditions, in like BG11, which is their regular growth condition, in a condition that mimics lunar soil, in a condition that mimics Mars regolith soil, and they get exposed, of course, in transit to space as well as on Earth to UV radiation. And then they take those, I guess they're desiccation resistant. So like they're not really growing in these spaces. They're just kind of sitting in this growth medium. And they take them out and they pick out a single cell. And that's, I guess, part of the useful technology here is that even if they have a really contaminated sample, they can verify what cell they sequenced because they have a microscope and optical tweezers. If people remember from our talking about remdesivir, the ability to trap something in using optical tweezers and move it, they can do that with single cells in this case, where they like rip off a cell from the chain of nostoc, and then they send that for sequencing. And the goal of the sequencing is to find mutations that occur, what mutations occur that make these, that in these sort of extreme conditions that could teach us something about where the adaptability lies within this species. Yeah.
Yeah. They say that
they find genes.
Sorry.
No, I just the TLDR is they find things.
Yeah, they find that. Go ahead. I think that there are specific genes that tend to get more mutations than others.
Yeah.
Yeah,
like the genes are in biofilm production. They find some genes maybe in some pigmentation. And again, they have versions that they didn't expose to didn't expose to space, but also didn't expose to radiation. And they're able to sort of parse away then what ones they think are coming from space specifically.
Yeah.
Like weird space things, which is probably gravity, right? Like that's a big part of being in space. And so both in the lunar and in the Martian soils, it's kind of interesting because they're going into like a lower gravity environment with a really weird nutritional balance. Like they really changed in some ways so many variables in the growth of these organisms. So what they find, being able to pick mutations out from that group is interesting because it sort of like relates to those wildly different conditions that this bacteria might find itself in.
Yeah, I mean, it's hard to like pull out anything more from that.
Yeah, I think that's everything.
Yeah, it's hard to figure out what the significance of those mutations are. I mean, why is one area? I mean, could it be that there's lots of copies of that genome or lots of other things? We don't know.
Yeah, it's super weird too because it's not like they're passaging. They're not like trying to evolve this bacteria in space. Like it's very much like the bacteria just had a extended visit into space and then it came down. So in some ways, the changes that they're seeing are places that genomic damage happens, right? Or like, and that doesn't get repaired properly or something like that. Because they see a lot of, they try to do a synonymous, non-synonymous mutations to try to pick out like what are the places that are changing versus what are the places that are not changing. And I don't exactly, what they picked out, so they picked out non-synonymous to synonymous mutations. And and for some of those genes, they do see more, they see more synonymous changes, meaning that they're just things that are getting repaired, not things that are like changing because of some adaptive capability. I don't know. I agree. Like the significance of what they pull out is kind of confusing, given that things aren't growing, given that that they don't have like a functional, they don't have any function associated with these things. Only the hypotheses of like knowing like these genes already, how they're annotated.
Yeah, it's some science that they did that happened, so therefore we need to hear about it. And because if you go to space, you want to make sure you get something out of it. Um, and we got this paper, which I guess maybe like in 100 years, we might find out what the significance of these results were, maybe. But
Yeah.
until then, we've got another paper.
Yeah, like, oh yeah, so like I'll read the piece of discussion that they have that, like, to me I'm like, okay, so how am I supposed to get something out of this now? Due to the
Okay.
anhydrobiotic conditions during our experimental setting, we do not expect that significant biological activity, replication, or DNA repair activities took place. Therefore, our results are most likely solely a reflection of DNA damage occurred during the experimental conditions, as has been verified by other research groups under similar settings. Right, so it's not talking about like the evolution of these organisms, right? Like, even though they set up, I feel, the paper, and I set it up too as I talked about it, like that there's some capacity for this organism to like live out in space and do these things. This is almost the most preliminary of tests, just to see like how damaged they got when they sent them up.
Yeah, I mean, it might just be
Uh-huh.
like a test of like what, because I know like with like say human genes, there was this instance where if you got histone like grabbing onto the human gene to protect it, then it's less likely to have damage. But if it's transcribed a lot, then
Hmm.
that's more likely to get damaged. So, for instance, if you got a thing like that, if you got genes that are very important, then they're more likely to be exposed and more likely to be damaged by
Hmm.
certain. And
Hmm.
so maybe that's something like that's happening. Maybe there are some kind of protective proteins that aren't present on these specific genes because they're kind of useful during the dormant state in space. Who knows?
Yeah,
Like
yeah, I
there's
kind of like that. Yeah, this could be insight into the dormant state of this organism more than anything, right? Just how it holds on, not like,
yeah,
yeah, mhm, how is it
like
holding on?
and that kind of would for me that would explain like say why the biofilm genes are important because those might be important during that kind of spiral. But it's this is like talking out into the air. I don't know what the this isn't. That's just like one hypothesis that came up in the 5 seconds that I've thought about this paper. So
Yeah, yeah, yeah. Yeah, yeah. Mutations occur even in this condition, though, right? Like, I guess that in and of itself is interesting. Mutations occur in this condition, and those mutations aren't changed. Like, they get repaired in some way, or like they only exist if they don't make significant changes.
well, yeah, that's another thing.
Um,
These mutations are based off of genomes, but the thing is like the radiation like it messes things up on a chemical level. So
mhm,
that might
mhm.
change the way things bind. So if you get like an adenine that's changed to a slightly different chemical that might bind in a way that's cytosine but isn't cytosine. It might be so we don't actually know the like full we don't know the chemical structures of the
Yeah.
genomes that have been changed. We're so there is like another layer but underneath this that we're not seeing.
Yeah, yeah, there could be an explanation. Yeah, there could be an explanation that maybe isn't even on the biology part of it, right? Or yeah, but I think the opening, I like that thought that like there are some genes that might be more exposed, right, in the survival
Hmm.
condition, and so maybe that makes them more likely to be mutated in some way. Anyways, that's this report. You can't let
Yeah,
space go. Like it takes a lot of resources to do this experiment. Yeah, and so now we know.
yeah. That And so now we've got our next paper, pervasive translation in Mycobacterium tuberculosis.
Hmm. Yeah, this is cool. I guess I haven't heard of pervasive translation before. Apparently, it's something that's been recently thought about in bacteria, where you can have translation. Basically, there's more open reading frames than you would think. Open reading frames meaning
Mm.
where mRNA is made, transcribed, and then where those mRNAs become small proteins. There's a lot more than people expect in bacteria based on like our predictive capabilities. And these authors take the techniques that were used to discover that originally, and they apply them to Mycobacterium tuberculosis. Um, and most often, the products of these pervasive translation events are small. So it also speaks to sort of the fact that we've focused a lot, I guess, would be based on our tools in molecular biology. We focus a lot on like larger proteins and bigger things, right? Like thinking like of these large complexes that do big important tasks, but there's probably also tons of small things that are doing small tasks in the background. And on the hypothetical side, the authors and people that work in this field also think about pervasive translation as a source of potential diversity for mutation and for adaptation, because you could have like a lot of small little products that aren't made, that not too many of them are made, but if they become useful in some circumstance, then maybe more of them can be made, and then potentially make that gene more important. Anyways, the method that they use to find all of these techniques is ribo-RET. It's a type of ribosome profiling, or ribo-seq, where they're sequencing things that are attached, sequencing the genes that are attached to ribosomes. But specifically, they use a drug of some sort called retapamulin, which arrests ribosomes around the start, wherever they started, wherever they landed, and clamp down on the mRNA. So that gives us like a great sense of like the start codons that go into things. And so using a combination of that ribo-RET as well as regular ribosome profiling, the authors are able to get a huge catalog of ribosomes that are trapped trying to make a protein, and a proportion of the ones that they find are things that have never been called ORFs before. So just they never got called by the algorithm to say like a gene is here making something. And so that's cool, right? I think that automatically like this paper is just a huge repository of data talking about stuff that we don't really think about being made sometimes, like we might discount, right? But actually there's a lot more being made inside of a bacterial cell. And that could be useful in a lot of different processes.
Yeah, I mean, I think that there is, because recently I was like talking to somebody about their work, which was on microprotein signaling, which was the idea that small proteins could form important aspects of signaling. And yeah, this kind of like, I think this kind of hones in on the fact that our technology is getting better at understanding what these small proteins are. Firstly, understanding these exist, and then start figuring out what they do. Because I mean, I think this paper does make an important point that they might do nothing, and they might just be like kind of chaff, like kind of a byproduct of the cell's production. But the thing is, we don't know, and it could. That's interesting. We always want to have that.
And I think what I'll bring out as like kind of a counterpoint, because I agree, like it could do nothing. I think it's very important to remember that there are lots of things in biology that like they don't have a function. They're around because they were part of a system before, or they're the beginnings of something that could be that could be important later, right? But right now they're just like a random piece of variation that isn't useful in any particular way.
Yeah, we've all had professors like that in school, right? Like they've been around for a while, but they'll come in useful when you want to find out where like the coffee cups are stored. I mean,
but I want to remind people that like there was a thought process on the genome previously, right? That said that so much of the genome doesn't make protein, so it's not important, right? And so like there is also the tendency to say that if we can't find what it's useful for, it might just be junk, right? And like, but that's not always the case as well. Like there's also useful things inside of those. There's also function to be found in those regions. I think the paper does well. Like you were earlier on a graph that I thought sort of just summarized all of their findings from their bioinformatics. One of the things I didn't point out was they also found isoform. They categorized a section of things they found as isoform, meaning that it had the same end codon, the stop codon as known open reading frames, but it had a different start one. So like this could mean like this means that there's more truncated versions of proteins that we already know floating around in a bacterial cell. That's sort of how I interpret that observation. And again, that is very interesting from the evolutionary potential standpoint, because because it is a protein that has some function potentially on a regular ORF, then maybe truncated versions could be useful in other in other ways, right? And if the cell has a way of like making more of those truncated versions, that could be adaptive in very specific scenarios. But yeah, but they validate everything. They validate a bunch of the things they find with mass spec and with electrophoresis. So like at some level too, what I like about, or not at some level, what I like about this paper is that it did this sort of ribo-seq, ribo-ret comparison. They did bioinformatics to pick out all these different pervasively translated ORFs, and then they validated with looking for those proteins. So they can feel a little bit confident that they're not picking up on stuff that doesn't become protein. Whether or not that's significant still remains to be answered, right? Like whether it has a significant biological function, but they are being made. It's not just like stuff that gets degraded away really quickly.
Yeah, and that's interesting because that means the cell is putting in resources for this, and the question is there a reason for that? And
yes,
yeah,
yes.
I mean,
yes.
So, and
okay, that's all. I think it's really cool. We'll keep it in mind. I'm sure it'll come up many more times as we read through microbiology literature.
Yeah, and that brings us to the end of our show. So that's all, folks.
Yeah. We normally say something when we go, right? Okay,
so join us next week for more cool microbiology news.
Yeah, we want to remind everyone that while we're still very enthusiastic about microbiology and somewhat qualified, it's possible that we didn't get everything right. Science about thinking critically and asking the right questions. So if you have any questions or corrections, just let us know in the comments.
I agree, and you can also reach out to us over Twitter at microdwjc. We both believe in peer review. We both believe that peer review is a process in which we can all participate in. So we hope you all have had a good time listening to us ramble on about microbiology today. If you think you have anything to add or have found something unclear, please let us know in the comments.
And we want to say that we're not going to be here in 2 weeks. We're taking a little vacation, and so we'll be back in a month.
Yeah, that's right.
Yeah, and hopefully, and hopefully in the month we might even have. We're gonna try to investigate like this deep dive in monkeypox that we've been talking about a little bit in the background. So hopefully that month will give us some time, but we'll see. No promise. It might be back with a news show.
Yeah. So it's a pleasure seeing you as our audience. Well, I'm not seeing you, but it's a pleasure to know that you're there. It's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content.
Not next week, a month.
Oh, a month away. Tune in a month. So that would be in November. Yeah. So, but yeah, it's been a pleasure. Bye.
Bye-bye.